Hello! In today's video, we shall learn in detail about biomolecules. By the end of this video, you'll have a clear understanding about different carbohydrates, namely monosaccharides, disaccharides, polysaccharides, lipids, proteins, deoxyribonucleic acid, ribonucleic acid, along with their molecular structure. Let us begin with the introduction to biomolecules. Biomolecules are organic molecules required for the growth and maintenance of living organisms. These molecules comprise of four elements, namely carbon, hydrogen, oxygen, and nitrogen. Carbohydrates, lipids, proteins, and nucleic acids are the four major types of biomolecules. The study of these biomolecules is known as biochemistry. First, let us learn about carbohydrates in detail. Carbohydrates are polyhydroxyaldehydes or polyhydroxyketones. They are composed of carbon, hydrogen, and oxygen atoms, and the general formula is as shown. Carbohydrates are called saccharides and can be classified based on the number of products obtained after hydrolysis. They can be monosaccharides with single unit of polyhydroxyaldehydes or ketones, oligosaccharides with 2 to 10 units of monosaccharides, and polysaccharides composed of several smaller monosaccharides. Based on the number of carbon atoms, monosaccharides may be triose with 3 carbon, example glyceraldehyde, tetrose with 4 carbon, example erythrose, pentose with 5 carbon, example ribose, hexose with 6 carbon, example glucose, and heptose with 7 carbon, example acetoheptulose. Depending on the number of sugar units, the oligosaccharides may be termed as disaccharides and trisaccharides with 2 and 3 sugar units respectively. When polysaccharides are composed of only a single type of sugar units, then they are known as homopolysaccharides. They can be further divided into structural polysaccharides, example cellulose in plants and chitin present in fungal cell wall, and storage polysaccharides, example starch in plants and glycogen in animals. When polysaccharides are composed of 2 or more different types of sugar units, then they are known as heteropolysaccharides. Example, heparin, an anticoagulant, hyaluronic acid, a lubricant, and murine present in bacterial cell wall. By the way, if you're getting some value out of this video, please like and share this video so that all of us can learn, unlearn, and relearn together. Also, please consider subscribing the channel and press the bell icon to get instant notification of all the upcoming videos. Now, let us continue with carbohydrates. Monosaccharides are the simplest group of carbohydrates and cannot be further hydrolyzed into simpler sugars. Glucose and fructose are monosaccharides with chemical formula C6H12O6. They are colorless, crystalline solids soluble in water. They have a single unit of polyhydroxyaldehydes or ketones. Aldehyde group is always present at carbon number 1 of the monosaccharide molecule. Such sugars are known as aldoses or aldose sugars. Ketone group is always present at carbon number 2 of the monosaccharide molecule. Such sugars are termed as ketoses or ketose sugars. Monosaccharides are known as triose, tetrose, pentose, hexose, heptose when the number of carbon atoms are 3, 4, 5, 6 and 7 respectively. Stereoisomerism is an important character of monosaccharides. Compounds having same structural formulas but different spatial configuration are known as stereoisomers. A carbon atom is said to be asymmetric when it is attached to 4 different atoms or groups. The number of asymmetric carbon atoms determines the number of possible isomers, which is equal to 2 to the power n. Glyceraldehyde with 3 carbon atoms is the simplest monosaccharide with 1 asymmetric carbon atom. It exists as 2 stereoisomers, namely D-glyceraldehyde and L-glyceraldehyde. Glucose has 4 asymmetric carbon atoms and therefore has 16 isomers. D and L isomers are mirror images of each other. Dextrorotatory isomer is the optical isomer that rotates plane polarized light to the right, while levorotatory isomer spins plane polarized light to the left of the asymmetric carbon atom. In D-glucose, the OH group is on the right side, and in L-glucose, the hydroxy group is attached to the left side of the asymmetric carbon atom. In the case of D-fructose, the OH group is on the right-hand side, and in L-fructose, it is on the left side of the asymmetric carbon atom, forming a mirror image of each other. They are known as enantiomers because they cannot be superimposed when placed over one another. Many monosaccharides exist both in open straight chain form and ring form. Cyclic forms can be furanose or pyranose with alpha and beta anomers. If the ring is 5-membered, it is called as furanose sugar. Sugar with 6-membered ring is called as pyranose sugar. Now, let us understand about disaccharides. Disaccharide sugar is formed when 2 units of monosaccharides are joined by a carbon-oxygen-carbon linkage known as glycosidic linkage. Disaccharides are simple sugars, crystalline, sweet to taste, and soluble in water. Sucrose or cane sugar, lactose or milk sugar, maltose or malt sugar are disaccharide sugars. Sucrose is a disaccharide composed of 2 monosaccharides, namely glucose and fructose. Its chemical formula is C12H22O11. The acetyl oxygen from hemiacetal of glucose is joined to the hemiketal of the fructose, forming sucrose. The linkage is formed due to the reaction of the anomeric carbon of one cyclic monosaccharide with the OH group of a second monosaccharide. Lactose is a disaccharide composed of one molecule of D-galactose and one molecule of D-glucose joined by a beta-1,4 glycosidic bond. Maltose is also a disaccharide formed from 2 units of glucose joined with an alpha bond between the first carbon atom of one glucose molecule to the fourth carbon atom of the second glucose molecule. Next among the carbohydrates are the polysaccharides. Polysaccharides are long chains of carbohydrate molecules composed of several smaller monosaccharides bound together by glycosidic linkage. They are also known as glycans. Polysaccharide chemical formula is as shown. Polysaccharides are categorized into 2 classes based on the type of monosaccharide units they contain. They may be homopolysaccharides or heteropolysaccharides. Homopolysaccharides are composed of only a single type of sugar unit. Example, cellulose, chitin, starch, glycogen. Depending upon their function, they are classified into structural homopolysaccharides, example cellulose, chitin, and storage homopolysaccharides, example starch, glycogen. Cellulose is a linear, unbranched polymer of glucose units joined by beta-1,4 linkages. In plants, cellulose helps in maintaining stability and provides rigidity to cells, organs, and organisms. Chitin is a natural polymer found in the fungal cell wall. It is a linear, long chain polymer composed of N-acetyl D-glucosamine residues joined by beta-1,4 glycosidic linkages. Chitin gives stability and rigidity to the cell wall of fungi. The exoskeleton of arthropods like lobsters and crabs are also made of chitin. Starch is a homopolysaccharide composed of D-glucose units joined together by alpha-glycosidic linkage. It is known as glucosan or glucan. It is composed of a water-soluble amylose 15 to 20% and water-insoluble amylopectin 80 to 85%. Amylose is a long unbranched polymer made up of 20 to 1000 D-glucose units bonded to each other through alpha-1,4 glycosidic bonds. Amylopectin is a branched polymer made up of many units of glucose bonded together by alpha-1,4 glycosidic linkages and alpha-1,6 glycosidic linkages. Starch acts as a reserve of energy supply. Glycogen is an extensively branched glucose polymer consisting of 5,000 to 30,000 glucose residues linked by alpha-1,4 and alpha-1,6 glycosidic bonds in linear strands and at junction points. Glycogen maintains glucose homeostasis in the animal body. Heteropolysaccharides or heteroglycans are composed of two or more different types of sugar units, example hyaluronic acid. This acid is a heteropolysaccharide composed of D-glucuronic acid and N-acetylglucosamine bonded by beta-1,4 linkages. It helps in reducing skin wrinkles and acts as a lubricant. Heparin is composed of repeating units of sulfated glucosamine and uronic acid and is joined by alpha-1,4 glycosidic bonds. It acts as an anticoagulant and prevents blood clotting. Next, let us learn about lipids. Lipids are organic compounds that contain hydrogen, oxygen, and carbon atoms. Lipids are compounds that are soluble in organic solvents like benzene, chloroform, and insoluble in polar solvents like water. Lipids include fats and oils or triglycerides, phospholipids, waxes, etc. Based on chemical composition, lipids are divided into three categories, namely simple lipids, example fatty acids, triglycerols, waxes; compound lipids, example phospholipids, glycolipids, lipoproteins; and derived lipids, example steroids. Fatty acids are monocarboxylic acids with long hydrocarbon chains. They are amphipathic molecules, that is, their carboxylic group are hydrophilic or polar, and the hydrocarbon chains are hydrophobic or non-polar. Fatty acids may be saturated or unsaturated. Saturated fatty acids are simple, unbranched, and linear chain of CH2 groups connected with a carbon-carbon single bond and one carboxylic acid at its end. Their general formula is as shown, where N represents the number of methylene group. Palmitic acid is a saturated fatty acid with a 16 carbon chain. Unsaturated fatty acids have one or more double or triple bonds. The general formula is as shown, where N can be any integer. Stearic acid is an unsaturated fatty acid with an 18 carbon chain. Cis fatty acids have two carbon bonded by a double bond, and their hydrogens are on the same side of the carbon atoms. Trans fatty acids have two carbon bonded by a double bond, and their hydrogens are on opposite sides of the carbon atoms. Fats and oils are esters with long chain carboxylic acids and long alcohols. They are called triglyceride because they are esters composed of 3 fatty acid units joined to glycerol. Glycerol contains 3 carbons, 5 hydrogens, and 3 hydroxyl groups. Fatty acids have a long chain of hydrocarbons with a carboxyl group attached. In a fat molecule, the fatty acids are attached to each of the 3 carbons of the glycerol molecule with an ester bond through the oxygen atom. Waxes are long-chain non-polar lipids. They are esters of fatty acids and long-chain alcohols composed of hydrocarbon chains of 14 to 36 carbons. Waxes form waterproof coating on the surface of plants and animals, example, beeswax, paraffin wax. Phospholipids consists of a glycerol molecule, 2 fatty acids, and a phosphate group that is modified by an alcohol. Phospholipids are amphiphilic. It has a hydrophilic head with a phosphate group and 2 hydrophobic tails obtained from fatty acids joined by an alcohol residue. The phospholipids derived from glycerol are called phosphoglycerides. Steroids are another class of lipid molecule with a fused ring structure. They are hydrophobic and insoluble in water. All steroids have 4 linked carbon rings. Cholesterol is a steroid with a short tail. Steroids with OH functional groups are classified as sterols. Next type of biomolecules are the proteins. Proteins are polypeptide structures made up of 1 or more long chains of amino acid residues. The structure of protein can be divided into 4 main categories depending on the level of complexity. They are primary protein structure, secondary protein structure, tertiary protein structure, and quaternary protein structure. Primary structure of protein is a linear sequence of amino acids bound together by covalent peptide bonds to form a polypeptide chain. These bonds are formed between the N-terminal and C-terminal of amino acids. Secondary structure is a folded structure of protein containing regions of amino acid chains that are stabilized by hydrogen bonds from the polypeptide backbone. These hydrogen bonds give rise to alpha helix and beta pleated sheet of the secondary structure. A coil formed by hydrogen bonds between carbonyl group and the amino group of different amino acids forms alpha helix. Beta pleated sheet is formed by hydrogen bonds between carboxyl group of one amino acid on one sheet and the hydrogen molecule of an amino acid on another sheet. Tertiary structure of protein is a 3-dimensional structure formed due to the interaction between the R groups of amino acids. When folding of polypeptide chain takes place, the hydrophobic R groups of non-polar amino acids are found in the interior of the protein, while the hydrophilic R groups are seen on the outer side. Hydrogen bonds, electrostatic or ionic bonds, covalent bonds, or hydrophobic bonds are involved in the formation of tertiary protein structure. Quaternary structure of protein is formed through the side chain interactions between two or more polypeptides. Each polypeptide chain is called a subunit. It is formed by bonds between R group of different amino acids within the polypeptide chains, which gives various shapes to the protein. Finally, let us understand the structure of nucleic acids. Nucleic acids are the building blocks of living organisms. They are the biopolymers of large biomolecules essential to all forms of life. Nucleic acids are long chain-like molecules composed of a series of identical building blocks called nucleotides. Deoxyribonucleic acid DNA and ribonucleic acid RNA are the two types of nucleic acids found in cells. Each nucleotide consists of three parts: a pentose sugar, phosphate group, and nitrogen base. DNA contains deoxyribose sugar, while RNA contains ribose sugar. One oxygen atom is absent from second position in deoxyribose molecules. Each sugar at the 3' and 5' carbon position contains phosphoric acid and forms the sugar phosphate backbone. The nitrogen bases in nucleic acids are heterocyclic organic compounds containing nitrogen and carbon in their rings. They are of two types, namely pyrimidines and purines. Pyrimidine is a 6-membered unsaturated ring compound with 4 carbon and 2 nitrogen atoms. 3 different types of pyrimidines are cytosine, 2-oxy-4-aminopyrimidine present in both DNA and RNA, uracil or 2,4-dioxypyrimidine present only in RNA, and thymine or 5-methyluracil present only in DNA. Cytosine contains one keto-oxygen at position 2 and an amino group at position 6. Thymine contains 2 keto-oxygen at position 2 and 6 and a methyl group at position 5. It is also known as 5-methyluracil. Uracil is similar to thymine but does not have a methyl group at position 5. Purine has a double ring structure. The purine nitrogenous bases are characterized by their single amino group at the C6 carbon in adenine and C2 in guanine. Adenine is a 2-ringed purine-derived nucleobase. It has an amino group attached at C6 position. In guanine, this position is occupied by a keto-oxygen. The other amino group is present at position 2. A combination of pentose sugar and a purine or pyrimidine base is known as a nucleoside. A combination of a nucleoside and phosphate group is known as a nucleotide. So, a nucleotide consists of a pentose sugar, nitrogen base, and a phosphate group. In a nucleotide, nitrogen base is attached to first carbon of the sugar molecule by glycosidic bond. The phosphate group is joined with the fifth carbon of the sugar by sugar phosphate bond. DNA is a molecule made up of two polynucleotide chains that coil around each other to form a double helical structure. It contains deoxyribose sugar, nitrogen base, and phosphate group. Purines are adenine and guanine, while pyrimidines are thymine and cytosine. Purine base always pairs with pyrimidine base. Adenine pairs with thymine by a double hydrogen bond. Guanine pairs with cytosine by a triple hydrogen bond. A DNA molecule is composed of two strands. Each strand is made up of nucleotides bonded together covalently between the phosphate group of one and the deoxy sugar of the next. The bases of one strand bind to the bases of the second strand with hydrogen bonds. The successive nucleotide of the same strand are linked by 3'5' phosphodiester linkage. RNA is a nucleic acid present in the nucleus as well as in the cytoplasm. They are of three types: messenger RNA, transfer RNA, and ribosomal RNA. RNA is a single-stranded structure. Chemically, it is a polynucleotide molecule. It contains a pentose sugar, a molecule of phosphate group, and a nitrogen base. Adenine and guanine are the purines, while cytosine and uracil are the pyrimidines. Adenine pairs with uracil with the help of two hydrogen bonds. Guanine pairs with cytosine with the help of three hydrogen bonds. RNA chain is formed by phosphodiester linkages between ribonucleotides. We have already learnt in detail about DNA and RNA in one of our previous videos. I will leave a link of the video. You can watch the video for complete understanding of DNA and RNA. So today, we have learnt in detail about four main types of biomolecules, namely carbohydrates, lipids, proteins, and nucleic acids. I have some practice questions for you. Please share your answers in the comment section below. You can pause the video and write down the questions if you like. Thank you so much for your time and participation. If you haven't subscribed to my channel yet, please consider subscribing the channel and press the bell icon to get instant notification of all the upcoming videos. Also, if you want to discuss any particular topic in biology, please mention that in the comments section below. I'll see you there. Goodbye. All the best.